Welcome back, everyone, to Chapter 7, the nervous system. And this is part 2 of the lectures for this chapter. And with this particular lecture, we're going to cover the communication of the nervous system. One of the basic components of the communication of a neuron is this concept of an action potential. And all an action potential is the change in the electric potential in the cell associated with the passage of an impulse throughout the entire cell. And that's all that means. That's an action potential. So let's go ahead and put this in a graphical format here. And so what we'll do here is let's further define it. Here on our x-axis, our y-axis is voltage in units of millivolts. And here on our x-axis we have time in milliseconds. And so when we look at neurons or any cell in the nervous system, it's in a negative resting state. And so what we mean by that is when we look at the voltage of a neuron at rest, its voltage is negative. And typically we see this at -7, anywhere between -70 and -65 for a normal neuron. But it can range from -40 up to -100 millivolts. But for our example here, we're going to say our resting state here in red is going to be -70. So I'm going to say this at rest. Now, for an action potential to occur, the neuron needs to become depolarized to the point where it'll propagate that action potential. And what we mean by that is the neuron needs to become more positive to a certain threshold. And we're going to denote that threshold line here in yellow. So what we mean by that is if the neuron becomes excited, right, it gets an outside stimulus. And we'll show this here in green for right now. If it does not reach this point of -50 millivolts, the action potential will not be generated. But if that stimulus reaches -55, all of a sudden the neuron will depolarize very quickly, and it'll reach a voltage of upwards to positive 40 millivolts. And so that's what we call, for this section, depolarization here in green. Once it reaches the apex at 40 millivolts, it will start going through the process of repolarization. And we're going to make this here in light blue. And so what we mean by repolarization is the neuron is becoming negative again. Oops, I cannot spell. Let's respell this here. Now, on the other side of this is once it reaches beyond its original resting potential of -70 millivolts, the neuron actually becomes a little more negative than it should. And it takes time to reach back up to that resting potential of -70 millivolts. And so this section here in yellow, this is termed the refractory period. Now, The refractory period can really be divided into two components. The first, I'm going to denote as one, is called the absolute refractory period. And what this means is no matter how much stimulation the neuron is occurring, it cannot generate another action potential, no matter what. The second portion, I'm going to denote here in green, is termed relative refractory period. And what this means is with the relative refractory period is an action potential can be generated with a greater than normal stimulus. That's all that means. In short, this is what an action potential is in graphical form from our definition. And so in practice, here I want to discuss on how an action potential is started here in the trigger zone and is transmitted through the axon of the neuron itself. So here we have the soma, here we have the axon, and here we have our synapse. Here we have our action potential graph. On the y-axis we have the threshold voltage, and here on the x-axis we have time in milliseconds. Here on the axon we have two different types of channels. The first in yellow are called leak-gated channels. And these are channels that are always open, allowing for the efflux of particular ions. Secondly, here in green, we have what's called voltage-gated channels. And these are types of channels that are open and closed depending on the polarity of the neuron itself. And so this threshold component that we talked about with the action potentials is important. This is the determining factor on when these voltage-gated ion channels open. And so when we have enough summation of excitatory stimuli that occurs in the soma right before the trigger zone, and it reaches this threshold value of negative 50 millivolts, it will open this voltage-gated ion channel. And in this particular case, these voltage-gated channels are sodium voltage-gated channels. And so what's going to happen is when these voltage-gated channels open, sodium is going to flow in, causing the membrane to become more and more positive, which is what we call depolarization. And as this particular voltage-gated channel becomes, and portion of the membrane becomes more positive, it's going to trigger the next voltage-gated channel to open to allow more sodium to come in and become more depolarized or positive. And the same with the next voltage-gated channel. And because of what's going on right here is here with the sodium-gated channels is we have such a high concentration of them here in the trigger zone. This is why we see usually an action potential generated here within the trigger zone itself. And so with this initial depolarization of these first grouping of voltage-gated sodium ion channels, what we see is this dramatic rise of depolarization within the membrane. And with this rise, the sodium is trying to reach its equilibrium value of negative 50 millivolts. And this should read right here, positive 50 millivolts. And this should read positive 40, not 20. So typically what we do see is that the neuron membrane does not reach that positive 50 millivolt range. It typically peaks out at a positive 40. And that's why we always see with our graphs, we see a positive 40 millivolts. And why do we see sodium stopping to come in, peak out at 40 millivolts is because the sodium-gated voltage, the voltage-gated sodium ion channels for sodium tend to close at higher voltages. So we tend to see them close at anywhere between 30 and 40 millivolts. And so what happens is we no longer see this influx of sodium ions when we start hitting this 40 millivolt value. And so when the voltage-gated sodium ion channels close at this, when they reach this 40 millivolt state, is they become inactive for a certain period of time. So no matter how much stimulation they are given, they will not reopen to allow more sodium in. And then as quickly as we see the sodium voltage-gated channels open and close, with the action potential itself, we start seeing the repolarization of the neuron itself. And when it repolarizes, it tends to go more negative than it needs to for its resting membrane, this resting potential at roughly negative 65 millivolts. And this is due to the result of potassium leaving the neuron. And this is done through a couple of different ways. Is first, potassium will leave through these leak-gated channels, which are always open. But on the other side of it too is the voltage-gated channels for potassium open up as well to help with the efflux of potassium itself. And by doing this with these voltage-gated channels, we can have more potassium flow out of the cell faster than with just the leak cells alone. The efflux of potassium will stop once it reaches a certain negative value. And this is due to the electrical forces at play with these particular ions. And so once the value reaches roughly at about negative 80 millivolts, these potassium voltage-gated ions will shut off, stopping the repolarization of the neuron itself. And so as these potassium voltage-gated ion channels begin to close, the permeability to potassium starts returning to normal, and that's where we start getting this slow gain back to our resting potential. And this kind of occurs through the leak channels for the potassium because our voltage-gated channels are closed at this point. And so this movement of sodium and potassium ions that creates this waveform for the action potential starts here at the trigger zone. And then as the influx of sodium and efflux of potassium occurs, this action potential in the same process occurs throughout the entire axon, and it kind of moves in waves. And so as that waveform, for depolarization and repolarization occurs, it moves down the axon, the body of the axon, until it reaches the synapse. And this entire four-step process of sodium in and potassium out and repolarization occurs at each point where there's these channels along the body of the axon. And as so as the action potential is propagated down the axon itself, we have, following right behind it, is we have this repolarization occurring. And so with this repolarization following behind the action potential itself, it actually stops the ability of any type of depolarization to occur behind it. And so this is the aspect that makes action potential propagation a one-way street. And so once the action potential reaches the synapse, we start moving into the point where the signal is transitioned from one cell to another. And the way the signal is transduced is through what we call neurotransmitters. These neurotransmitters are found in vesicles in the synapse itself. And so I'm going to draw all these here. And all vesicles are just little storage sacs that are found in cells that hold certain biochemicals. In this case, we're going to be holding different neurotransmitters. And so what happens with these neurotransmitters once the action potential reaches the synapse is that these vesicles then start moving towards the presynaptic terminals. And once these vesicles reach the membrane, the neurotransmitters are released into the void between neuronal cells. And these are in this region is typically termed the synaptic cleft. And so what happens is these neurotransmitters flood this area and flow until they bind to their particular receptors in the dendrites. And these receptors in the dendrites have a term called ligand receptors. And then so what happens is once you have a certain concentration of these neurotransmitters bonding to their particular receptors, that signal is then transmitted through excitatory summations to the trigger zone, and thus another action potential begins. And that's how neuronal cells transmit action potential from one cell to another. So with this particular lecture, we covered a couple of different areas. The first being is what an action potential is. And we graphed its parts. Secondly, we talked about how an action potential is transmitted within a neuron itself. And lastly, we briefly covered how neurons communicate with each other. So if you can understand these steps, that is the crux of this part of Chapter 7. I hope this helps. Take care and have a great day.